Good day everyone, my name is Nico Nathaniel Esofeta, and in this video, I'll be showing you a stop-motion demonstration of meiosis. But before that, I'm going to briefly explain what meiosis is.
Meiosis is a type of cell division in sexually reproducing organisms that occurs to produce gametes, or the sperm and egg cells. It includes 2 rounds of cell division that will lead to 4 daughter cells, each with only 1 paternal and maternal copy of a chromosome. In meiosis, there are a total of 2 phases, both having 4 stages each. Each of these phases and stages will be shown in the stop-motion demonstration. Now without further ado, let's get into it. The first part of the demonstration occurs before meiosis, known as the interphase. Interphase is where the chromosomes of our male and female cell duplicate in preparation for the next stage. Now we are on to the first phase of meiosis. The first phase starts with prophase 1, where the complex of DNA and protein, known as chromatin, condenses to form chromosomes. A large structure called the meiotic spindle also forms from long proteins called microtubules on each side, or pole, of the cells. Between prophase 1 and metaphase 1, the pairs of homologous chromosomes form tetrads. Within the tetrad, any pair of chromatid arms can overlap and fuse in a process called crossing over or recombination. Now that prophase 1 is complete, the 2 cells can proceed to the next phase. The next phase is called metaphase 1. In metaphase 1, the homologous pairs of chromosomes align on either side of the equatorial plate. The tetrads of each cell line up at the center of the cell in preparation for anaphase 1. After metaphase 1, anaphase 1 follows. Anaphase 1 is where the homologous chromosomes are separated and pulled into the opposite poles of each other. What follows is telophase 1, wherein the chromosomes are enclosed in a nuclei. The spindle fibers also disappear during this stage. The cell now undergoes a process called cytokinesis that divides the cytoplasm of the original cell into 2 daughter cells. With the first cell division complete, there are now 2 cells that are now ready to undergo the second phase of meiosis. At the beginning of meiosis 2, the cells undergo prophase 2. During prophase 2, the chromosomes condense and a new set of spindle fibers forms. The chromosomes begin moving toward the equator of the cell. What follows is metaphase 2, where the centromeres of the paired chromatids align along the equatorial plate in both cells. Then in anaphase 2, the chromosomes separate at the centromeres. The spindle fibers pull the separated chromosomes towards each pole of the cell. Finally, during telophase 2, the chromosomes are enclosed in nuclear membranes. Cytokinesis follows, dividing the cytoplasm of the 2 cells. After meiosis, 4 haploid daughter cells go on to develop into either sperm or egg cells.